Hi everyone, I thought I would make this really quick video on how restriction enzymes work to refresh you on that animation I showed in class. So a DNA palindrome is a sequence of DNA bases that can be read left to right and right to left, and they say the same thing, quote unquote. So if we watch the animation, you see that the sequences reading left to right on the top strand, right to left on the bottom strand are the same. AT, GCC, GGCAT. The restriction enzyme recognizes this particular palindrome and attaches to the DNA. Every restriction enzyme has its own palindrome it recognizes. The restriction enzyme then snips the DNA backbone in a specified location that's specific to that particular enzyme. Just like the palindromes are unique for each enzyme, so are the places where they snip. So this one that I made up just happens to snip between the first A and T. The enzyme detaches and the DNA strands separate, exposing sticky ends that can bind to their complements. So the T, G, C, C, G, G, C, A are nucleotides that are ready to hydrogen bond with their complements. T is ready to go with A, G with C, C with G. And if another sequence of DNA has the same sequence, the same type of sticky end, they can bind very easily. They do it spontaneously because they just fit together. That's how their geometry works. So when you have a gene of interest, the pink in the middle, you treat it with the same restriction enzyme that has the same sticky ends and are ready to bind with the DNA molecule we treated originally. So they will move together spontaneously and bind with hydrogen bonds between the bases. Finally, once these bases are hydrogen bonded to one another, an enzyme called DNA ligase comes in, and it will use ATP, the energy currency of the cell, to bind the backbones back together to make them one solid strand of sugar phosphate, sugar phosphate, sugar phosphate, all the way down, and that completes the creation of the recombinant DNA. So if we do a quick recap, the DNA has a palindrome. The restriction enzyme recognizes that palindrome and binds to the DNA. It snips the DNA at a particular place and separates the strands. The sticky ends recognize other sticky ends that hopefully has a gene of interest, and then DNA ligase will seal those backbones back up again. Anyway, hope you enjoyed that quick tutorial.